I'm a bud tender at Havahart. I also have a background in science, and I just wanted to talk a little bit about the endocannabinoid system and why it's important. So, why is it important? Phytocannabinoids their health benefits, their medicinal benefits would be non-existent were it not for their interaction, within the endocannabinoid system. And so this is a very rough diagram, diagramming the accumulation of certain receptor sites, CB1 mostly in the central nervous system, peripheral, and CB2 in the immune system, but they are scattered throughout the body. I'm just going to talk basically about the elements that comprise this system, certain functions, and certain methods of studying the system. Now, Michelle knows much better than I do, I think, about this because she's actually, my background's in mouse genetics. Yours is, actually in this. So anyway, so this crude model demonstrates a lock and key, which is a really basic way of thinking about CB1 and CB2 receptors, which are not the only receptors within the endocannabinoid system, basically. But the basic idea is that you have a receptor sticking out of a cell, and the molecule that binds to it, like THC or an endogenous cannabinoid like an anandamine, binds to it, and this basically within the cell starts a signal transduction cascade. A lot of chemicals going crazy, a lot of molecules, genes being activated, among other things. And so the system itself is a lot more complex than just the receptors and the interaction between either phytocannabinoids like THC or endogenous cannabinoids like anandamine. So just a quick question. There are three elements really that comprise this system. Does anybody know or have any idea what they are? Yes.
I was just giving you a hard time.
No. Well, this is what I've found at least. You have endogenous cannabinoids, for example, anandamine, and then you have the enzymes that synthesize and degrade these cannabinoids. And then you have cannabinoid receptors, which, for example, are CB1, CB2, AA, and 2AG. And those are really the three elements. But why is it called a system? That I think is a good question. What comprises a system? Why is it a system in and of itself?
And all of them working together to make it work, make it happen.
Exactly. I mean, you've heard probably the phrase "the whole is greater than the sum of the parts." Well, entourage effects, among other things, with a number of terpenoids, really lead to health benefits. And this is, a lot of this research is cutting edge, and it is difficult to tease out, the interactions among cannabinoids in this system and how they actually contribute to health improvement. A common technique in molecular biology has been the reductionistic approach to look at one gene or one enzyme and see what that does. But in this case with the cannabinoid system, and tell me if I'm wrong, it's difficult at least to tease out one gene, say it might be helpful, but you need to look at that gene in the larger context of the system.
Yeah, I'm not a geneticist.
Okay. All right. You're being kind. So these are a few functions of the endocannabinoid system. The ability to generate new neuronal cells by sparking progenitor cells that then allow these cells to be synthesized, causing cell death in damaged cells, also called cell apoptosis. That is important in cancer, and we'll talk a little bit more about that. Stimulating appetite. There's inverse correlation between leptin and CB1, CB2 receptors, and mice actually that have had their receptors, CB1 receptors, knocked out, their appetite decreases. So this indicates that the accumulation of receptors of that type actually stimulates directly appetite. The treatment in preventing of seizures, treating a variety of other illnesses, chronic conditions, and diseases in ways that we have yet to understand. And you can add to this list, significantly, honestly.
So, Bill, when you say it's generating neuronal cells, is that brain cells?
Yeah. Yeah.
Nerve tissue?
That's
For
right.
memory.
Yeah. Yeah, that's right. I mean, you could speak more on that than me, Michelle, actually, I'm sure.
Again, that, regenerative medicine isn't something I studied in depth either. There probably is data out there. I know that, well, outside the examinoid system, as an aside, CBD has been shown to stimulate the cells that wrap neurons or myelin.
Okay, the myelin
so
sheath.
Relevant dendrocytes.
Yeah, yeah.
But, all neurons have the cannabinoid receptor, and I'm sure the stem cells do as well.
Yeah.
But I think you're right that there has been some studies showing stimulating migration of stem cells.
Yes, yes. So methods of studying the endocannabinoid system. I'm most familiar with animal models. For example, researching cannabis and memory extinction. That's a very interesting line of study. So animal models, we'll talk about those in a little bit. Tissue culture methods, example researching the anti-tumor effects of cannabis. And then human studies, statistical population studies. So example, monitoring populations of individuals who use cannabis to treat illnesses ranging from cancer to arthritis. I think it's important to know just various methods of studying this due to the fact that the research that you draw from, say, I don't know, Wikipedia or other accumulations, it is important to tie it back to the actual studies that generate the results that you're familiar with. I mean, empirical or statistical, it's important to know the difference, honestly. And I'm going to tie all of this back to sort of common basic bud tender knowledge and how this can help improve, bud tender patient interactions. So, for example, post-traumatic stress disorder, THC and memory extinction. This is a very interesting line of study that I got interested in a little while ago. This also demonstrates that it's important to know if a patient comes in and says, "Look, I have post-traumatic stress disorder. What would you recommend?" Well, it would be very helpful to know certain symptoms of post-traumatic stress disorder. That would allow you to direct your attention to certain medicine that will help them. So, for example, one symptom is recurring memories of this traumatic event that are adverse or that interrupt one's life, basically, these memories, and they can cause night terrors, a lot of things going along with that. And, a common conception of cannabis is that it impairs memory. Well, in this situation, it's actually beneficial. So, THC, through binding to CB1 receptors, which are primarily in the brain, synapses and everything, suppresses the process necessary for the formation and long-term storage of memories, leading to enhanced memory extinction. Well, what does that mean? THC actually allows for these memories, these traumatic memories, or it facilitates these memories to extinguish themselves. And so someone walks in, and says, "I have PTSD. What would you recommend for me?" Well, given this second piece of data, that CBD actually slows down the effects of THC on the hippocampus, meaning that CBD actually interferes in certain ways with this memory suppression that THC is able to achieve. What would you suggest? What strain?
Indicas.
What's that?
Indicas.
High THC.
High THC, low CBD.
Yes, high THC, very low CBD. That's exactly right. That's what you would want to suggest.
Clear.
Yeah. And here's how rigorous this study is, which is interesting. So you have mice with CB1 receptors in the hippocampus, and then what is the control group? Knockout mice not possessing these receptors. So what are knockout mice? Very quickly. So knockout mice are mice that have had this CB1 receptor knocked out. You take an embryonic stem cell, and you've got the stem cell, and then you have the nucleus inside with all the genetic material, and you actually inject genetic material straight into that nucleus. It integrates with the other genetic material in there, and you can knock out certain genes like CB1. basically, or the gene that produces these CB1 receptors. And so as these cells divide, all of them have that same genetic makeup, basically a makeup that doesn't allow mice to produce these receptors. And so you come up with a line of mice that don't have CB1 receptors. And what they demonstrate is an impaired ability to erase traumatic memories. So that's a very strong, piece of evidence for the fact that THC working on CB1 receptors suppresses this process of memory formation. Or I'm sorry, suppresses memory formation. And so there's one concrete example of why, understanding a little bit more about these studies will help direct you as a bud tender toward the correct medicine to give to patients with chronic situations. Here I've run across a couple tissue culture studies, and this just means that you have a petri dish and you basically are growing, certain groups of cells, tumors, among other things. And so this isn't exactly the endocannabinoid system, but it's directly related. This tissue culture study, and I can provide all of the papers and everything that this is based off of too, so you know that I'm not just talking out of my ass. Which I still might be, but tissue culture study indicates that cell apoptosis, which is just programmed cell death, actually is achieved through cannabinoid interaction or is enhanced. One problem in cancer cells is that when they're impaired or when they're damaged, they do not die. And many cells are programmed to commit cell suicide when they find themselves damaged or compromised. And so that's one problem with cancer cells. They continue to proliferate even though they're damaged. And so another tissue culture study indicates that cell cycle arrest is achieved through cannabinoid action. What does that mean? Well, with phytocannabinoids, cannabinoids that occur in plants, but also endogenous cannabinoids inside our body that are manufactured there, like anandamide, they can actually affect the cell cycle such that in cancer cells, for example, cell cycle is just basically on hyperdrive. These cells are dividing so rapidly, and it does have to do in part with certain problems that have occurred in the cell cycle. And cannabinoid interaction can arrest this cell cycle and arrest actually mitotic cell growth. And then the last tissue culture study indicated that blood vessel growth in tumors was inhibited by certain entourage effect, an interaction, they think, of a number of different cannabinoids. And so basically this is like sort of strangling the tumor because blood vessels are needed to nourish this huge mass of cells that haven't paid attention to certain density stipulations and everything like that because they're damaged. And so if you strangle the cell, by cutting off blood flow, then the tumor shrinks, basically. And why is this important? Well, I think it is important to know why cannabinoids have certain anti-tumor effects, concrete. And these are a couple of reasons why. This just adds to an enriched understanding of why the endocannabinoid system is helpful in anti-tumor areas among other things. And that is about it. That's all I have to say.